Hello and welcome everybody to my channel Omni Bio Entrance, and today we will be discussing Kerala SET Biotechnology question paper conducted in the year July 2024. If you are new to my channel, please do like, share and subscribe. If you want still previous year's question papers of Kerala SET Biotechnology, you can go to my channel Omni Bio Entrance. In that playlist you see for Kerala SET exams, and there you will find. The energy value of a biogas is typically, so energy value, it refers to the amount of energy contained in the biogas, and in biogas it depends on methane content, carbon dioxide content and other gas content. So the answer here is 400 to 700 BTU per feet cube. Answer is option B. The excess of which of the following compound in the drinking water cause blue babies? It is NO3 minus. Answer is option C, that is nitrates. Then which of the following is not an algorithm for building phylogenetic trees? It is bootstrap. Bootstrap, it is not an algorithm, it is a resampling method and is used to evaluate phylogenetic tree stability and estimate confidence in tree topology. What is maximum parsimony? It finds tree requiring minimum number of changes. Neighbor joining, it constructs tree based on distance matrix, and maximum likelihood finds trees maximizing probability of observing data. Then next one. The eukaryotic RNA polymerase which is insensitive to amanitin is RNA pol1. It synthesizes 28S, 18S, 5.8S rRNAs. Then RNA polymerase 2, it is mRNA synthesis and is highly sensitive. Whereas RNA polymerase 3, that is tRNA and 5S rRNA synthesis, it is moderately sensitive. So the answer here is option B. Then which of the following is not a part of the regulatory framework of recombinant DNA research? Is IAEC. It is Institutional Animal Care and Use Committee. GEAC is Genetic Engineering Approval Committee. RDAC is Recombinant DNA Advisory Committee. IBSC is Institutional Biosafety Committee. So these three ABC are part of regulatory framework of the recombinant DNA research, whereas IAEC is not. The technology to clean up the pollution generated by other technology is called end of the pipe technology. So it refers to pollution control measures implemented after pollution generation. It focuses on it focuses mainly on treating or removing pollutants from waste streams. And examples include air scrubbers, wastewater treatment plant, emission control systems. Now what is front of the pipe? Front of the pipe is a preventive measure to minimize pollution generation. Cleaner technology is sustainable environment friendly approaches to reduce pollution. That is the use of renewable energy, green chemistry, etc. Identify the correct statement. COD is always higher than BOD. Yes, that's correct. Because COD values is the amount of oxygen that will be consumed by the chemical breakdown of both organic and inorganic compounds. That is COD. That is the amount of oxygen that will be consumed by the chemical breakdown of both organic and inorganic compounds. And BOD is the amount of dissolved oxygen needed for microorganism to degrade organic matter under aerobic conditions. That is BOD. COD values are always greater than BOD because COD considers both biodegradable and non-biodegradable substances and measures the total oxygen demand they require. While BOD, it measures oxygen demand of only the biodegradable materials over a limited time. Name the component of milk which is an excellent emulsifying agent. It is casein. Gelatin is derived from collagen used as a gelling agent. Then lactulose, it is synthetic disaccharide used in pharmaceuticals, and saccharin is an artificial sweetener, not a milk component. Then next one. The microorganism which favors health benefit is called probiotic. Prebiotics are a non-digestible food ingredient that stimulate growth of beneficial microorganism. Example of prebiotic include dietary fiber. An adjuvant is a substance that enhances immune response to a vaccine or an antigen. That is an adjuvant. Then microorganisms indigenous to a given ecosystem are called autochthonous. Answer is option C. Example, gut microbiota, coral reef microorganism, etc. They are indigenous to a given ecosystem. Then allochthonous is they are non-native and are introduced from outside of the ecosystem. Autogenous means they are self-generated or produced within an organism. And zymogenous means they are related to fermentation or enzyme activity. An aerobic method used for wastewater treatment is trickling filter. It is an aerobic method. They have asked for aerobic method. USAB is upflow sludge blanket anaerobic treatment. So it is an anaerobic method. USSB is upflow sludge bed anaerobic treatment. Again an anaerobic wastewater treatment. So that's also not the answer. Septic tank is also an anaerobic treatment. So C is also not the answer. Then next one. Method for the analysis of GM foods, it is by PCR. Enrichment culture is used for microbiological analysis. FTIR is Fourier transform infrared spectroscopy used for chemical analysis, not for GM detection. So what is the answer? PCR. Origin of bagel bread is in Poland. Answer is option D. Which among the following is a biodegradable plastic? It is PHA. A question similar to this was asked in January 2024 also. So PHA is polyhydroxyalkanoates produced by fermentation of sugarcane, potato starch and other biomasses. The organism which follows associative nitrogen fixation. So associative nitrogen fixation, the nitrogen fixing bacteria, they live in close association with the plant. And they will not form root nodules and they will not reside inside root nodules also. These bacteria, they fix atmospheric nitrogen and make it available to the plant, while the plant provides carbohydrate and other organic compounds. So azospirillum is a free living nitrogen fixing bacteria and associate with plant roots for fixing nitrogen. Acetobacter is also a free living nitrogen fixing bacteria. Rhizobium form root nodules. Anabena is cyanobacterium found in symbiotic nitrogen fixation. So the answer here is azospirillum. The metal present in nitrogenase enzyme is molybdenum. Then type of inhibition where the inhibitor binds to the enzyme. Whether or not the enzyme has already bound the substrate. Okay, it is not concerned whether substrate is bound to the enzyme or it is a free enzyme, that is without the bound substrate, but has greater affinity for substrate bound enzyme. Even though it can bind to the free enzyme, it has got greater affinity for the substrate bound to the enzyme. That enzyme substrate complex, it has got greater affinity. What type of inhibition it is? Is it competitive inhibition? No. Because here the inhibitor competes with the substrate for the active site. And inhibitor has greater affinity for the free enzyme, not for enzyme substrate bound complex or free enzyme. Then uncompetitive inhibition, here enzyme uncompetitive inhibitor bind to enzyme substrate complex only, but not to free enzyme. Here it binds to both enzyme substrate complex as well as free enzyme. So it is not uncompetitive inhibition. Non-competitive inhibition, a type of enzyme inhibition when inhibitor bind to an enzyme at a site other than the active site. Okay, that is non-competitive inhibition. Mixed is a type of inhibition that occurs when inhibitor bind both to the enzyme and the enzyme substrate complex. That is, it has affinity for whether the enzyme is free without a bound substrate or whether the enzyme has a bound substrate. So the answer here is option D, that is mixed inhibition. The absorption maximum of a red colored solution may be in the range of 500 to 600 nanometers. Specific binding of the substrate to the active site of trypsin is facilitated by residue in the specificity pocket. So which residue in the trypsin's active site is present, which facilitate the specific binding of the substrates of trypsin. Now what are the substrate of trypsin? They are basic amino acid, lysine and arginine. So C cannot be the answer. So it should be something which is, they are basic, lysine and arginine are basic amino acids, so they have a positive charge. Whereas aspartic acid, if you see, it is having a negative charge. So it will interact with this lysine and arginine. So chances of aspartic acid being present in the specificity binding site of the trypsin will enhance its binding ability to lysine and arginine. So answer is option A, aspartic acid. In solution, majority of fructose exists in. So it is the most stable form in solution is beta pyranose form. Then vanaspati is formed from vegetable oils by hydrogenation. You are learning this from your +2 onwards. Then when NADH and NADP plus ratio is high, which of the following metabolic pathways can take place? So high NADH and NADP plus ratio means the cellular energy state is reduced. There is increased reductive power and decreased oxidative phosphorylation. So which are the metabolic pathways that take place will be ethanol fermentation, fatty acid synthesis and cholesterol synthesis or lactic acid formation. So C can take place. Whereas glycolysis, Krebs cycle, fatty acid oxidation, oxidative phosphorylation, gluconeogenesis will not take place. A protein involved in electron transport, plastocyanin, ferredoxin, cytochrome C, all these are involved in electron transport. Find out the enzyme which is not a part of the multi-enzyme complex. Alpha ketoglutarate dehydrogenase is multi-enzyme complex. Alcohol dehydrogenase is not a multi-enzyme complex. It catalyzes the conversion of ethanol to acetaldehyde. At pH above isoelectric pH, the protein has. So if pH is less than Pi, the protein has positive charge. If pH is equal to Pi, then it has no charge. And if pH is greater than Pi, it has a negative charge. So at pH above isoelectric Pi, it should be, it shouldn't be pH, it should be Pi, it's a printing mistake. The protein has. So pH is greater than Pi, so it will have a negative charge. Answer is option B. The solubility of fatty acid in water is reduced when the longer fatty acyl chain and fewer the double bonds. So longer fatty acyl chain, they interact more strongly with each other, thereby reducing water interaction. Then fewer double bonds again result in the presence of more saturated chains. And saturated chains pack more tightly, forming crystals and thereby exclude water. So solubility of fatty acid in water is reduced when the longer fatty acyl chain and fewer double bonds. Fewer double bond means more saturation. Digestion of the stomach starts by the action of gastric juice. B. The major type of glycoprotein in eukaryotes is N-linked glycoprotein. B. N-linked glycoprotein where carbohydrate is attached to the asparagine residues. O-linked glycoprotein, carbohydrate attached to serine threonine residues. GPI-linked glycoprotein is glycophosphatidylenosytol anchor. Then next one. The functional unit of muscle fibers are known as sarcomeres. What is sarcolemma? Sarcolemma is muscle fiber plasma membrane. So that is sarcolemma. Sarcoplasm is muscle fibers cytoplasm, and sarcoma is a type of cancer. So sarcomeres is the answer. Then two phosphatidic acids are present in cardiolipin. It is composed of two phosphatidic acid groups, glycerol backbone and 4 fatty acid chain. Whereas lecithin, it is composed of phosphatidylcholine, single phosphatidic acid group. Dipalmitoyl lecithin, it is synthetic lecithin with palmitic acid chains. Plasmalogens, they are phospholipids with vinyl ether linkage, not two phosphatidic acids. Then an enzyme that catalyzes stereospecific isomerization. So stereospecific isomers, they have the same molecular formula, bond sequence, etc. But the three-dimensional spatial arrangement of atoms differ. As you can see here, this is citrate and isocitrate. See here, OH and H, it is different in isocitrate, but they are stereoisomers. They are isomers, and its reaction is catalyzed by aconitase. So answer here is option D, aconitase. Phosphoglucoisomerase, it catalyzes conversion of glucose 6-phosphate to fructose 6-phosphate. That is aldose ketone isomerization, not stereospecific isomerization. Bisphosphoglycerate mutase, it catalyzes conversion of 1,3-bisphosphoglycerate to 2,3-bisphosphoglycerate. It is phosphogroup transfer, not isomerization. Alanine racemase, it catalyzes racemization of L-alanine to D-alanine. So answer is aconitase. Then isozymes differing. Isozymes, they are different forms of an enzyme. That is like lactate dehydrogenase. There are 5 forms, LDH1 to LDH2. Then you have got hexokinase, that is from forms 1 to 4. So there are the different forms of an enzyme that catalyze the same chemical reaction but differ in physical and kinetic parameters. So isozymes differ in electrophoretic mobility, structure, kinetic parameters, all of these. Answer is option D, all of these. Then one of the reasons for the high energy hydrolysis of ATP is resonance stabilization of products. Answer is option B. The products of ATP hydrolysis, that is ADP and PI, are more stable, have more stable resonance structure than ATP. So answer is option B. Identify the correct statement about oxidases. So oxidases catalyze oxidation reduction reactions which involve transfer of hydrogen from substrate to oxygen to form water or H2O2. So they have asked for the correct statement. Perform their activity only in the absence of metal ions. That is not correct. Do not incorporate oxygen into the product. Yes. They transfer hydrogen, not oxygen, from substrate to they transfer hydrogen from a substrate to oxygen to form water or hydrogen peroxide. Incorporate one oxygen atom into the product. That is in the case of monooxygenases. They are that is oxygenases are enzymes that incorporate oxygen into the substrate. And there are 2 types, that is one that incorporates one oxygen atom into the product, that is monooxygenase, and incorporate 2 oxygen atom into the product, that is dioxygenases. So C and D are oxygenases, they are not oxidases. Then next one. Cholesterol lowering drugs such as statin hinders with the action of HMG-CoA reductase. So statins, they are competitive inhibitors of HMG-CoA reductase. HGPRT is involved in purine metabolism. HRP is horseradish peroxide used in laboratory analysis assays, etc. HMG-CoA synthase again involved in cholesterol synthesis pathway. Then synzymes can be developed by. So synzymes, they are artificial molecule designed to mimic enzymic activity, structure or function, and they are created using designing and synthesizing enzyme mimicking structures, yes. In vitro evolution, yes. Molecular imprinting, yes. So all of the above is the answer. India is not a signatory of UPOV, that is International Convention for the Protection of New Varieties of Plants. It is not a signatory. It has got its own sui generis, that is unique system, that is PPVFR, that is Protection of Plant Varieties and Farmers' Rights Act 2001. So as per this act, it protects plant varieties, recognizes farmers' right and encourages biodiversity conservation. WIPO is World Intellectual Property Organization, PCT is Patent Cooperation Treaty, and GATT is General Agreement on Tariff and Trade. What is the plant variety protection used in India? I told you just now, it is sui generis. What is the meaning of testing of the hypothesis? It is a rule for acceptance or rejection of the hypothesis of the research problem. Answer is B. Then the method used for the prediction of three-dimensional structure of a protein from known structures. Okay, you know, from known structures of one or more related proteins. That is called as homology modeling. Multiple sequence alignment is used in homology modeling, but it is not the most apt answer. Apt answer is homology modeling. Phylogeny is study of evolutionary relationship between organisms. Docking is it predicts the protein ligand interactions, not the three-dimensional structure of proteins. So answer here is homology modeling. Then if the value of mode and mean is 60 and 66 respectively, then the value of median is 64. Okay, median is equal to mode plus mean by 2. So 60 plus 66 is equal to 63. That is approximately equal to 64. So A is correct. But this formula is not the correct reason. Formula is mode plus mean divided by 2. So the answer here is A is correct, R is incorrect. Answer is option C. Then next one. The data type which is supported in C++ but not in C. It is Boolean data or bool, B-O-O-L. Whereas others, int, double, float, all these you can find both in C and C++. Which of the following technique is an analysis of the relationship between two variables to help provide the prediction mechanism? So it is regression, that is helping to provide the prediction mechanism. Standard error measures variability, not prediction mechanism. Then correlation measures strength and direction of the linear relationship between two variables, but it doesn't provide a prediction mechanism. Then next one, which one of the following statement is not correct? Bar diagram is one dimensional diagram. Yes. The bars in histogram touch each other. Yes. With the help of ogive curve, one can predict percentiles. Yes. Both line diagram and pie diagram are two-dimensional diagrams. Pie diagrams are two-dimensional, but line diagrams are one-dimensional. So that is not the correct statement. So answer is option D. Computer understands only binary language. Answer is option C.